Next major topic is polymerase chain reaction or PCR. Hopefully you have the basic background of this topic, and you may have studied this topic in other subjects that fall within the domain of bioinformatics, as it is also the part of syllabus of gene manipulation and genetic engineering. So we will also focus on this topic, and hopefully you may learn some new points when we will go through this major headings, that is polymerase chain reaction or PCR. So as far as its definition is concerned, it is the technology where we can amplify a few copies or even the single copy of the target DNA to a great order of magnitude, so that finally this technology, it can generate millions of copies that subsequently can be used for different purposes, like for cloning or even for sequencing purpose. So within PCR, we will first focus on the basic reaction of the PCR. As far as its basic reaction is concerned, तो इसमें जो बेसिक रिक्वायरमेंट है कि हमारे पास टारगेट डीएनए होना चाहिए, जिसको हम टेम्पलेट डीएनए कहते हैं. इसके अलावा वी आल्सो नीड अ प्राइमर विच इज़ अ शॉर्ट ओलिगोन्यूक्लियोटाइड, और ये वो पॉइंट है जहाँ से डीएनए की जो सिंथेसिस है, चाहे इन विवो हो या इन विट्रो हो, वो इस पॉइंट से सिंथेसाइज होता है. और इसके अलावा हमें DNA polymerases required होते हैं that can synthesize the copy of the template according to the order of the bases. So in basic reaction, first we need template that subjecting to heat denaturation step, it is denatured and converted into single-stranded DNA, and then primers, they bind at 3' ends, and then the DNA polymerases, it can guide the synthesis of complementary strands of the template or the target DNA. So this is the basic reaction of polymerase chain reaction or the PCR. So by using this technique, we can produce millions of copies of the target DNA, and this reaction can be explained diagrammatically in more detail. Here we can start with single segment or fragment of the DNA that we want to amplify, and within this single fragment, which is double-stranded, we have a specific locus that we have targeted for sequencing. So the region of target DNA to be amplified, it is indicated in yellow color, and then it is exposed to heat so that double-stranded DNA is converted into single-stranded form, and then other requirements like nucleotide bases and primers, they are added, and temperature it is also managed for this step. And after the primer attachment sites here, the DNA synthesis, it is followed. So first cycle, it resulted in the synthesis of 2 strands of the DNA. In the next slide, from 2 DNA double-stranded fragments, they are the substrate for the next reaction or for the second reaction. So there is also, we can repeat step 1 and 2. So double-stranded DNAs, they are converted into single-stranded form. So 4 single-stranded fragments now they are available for further reaction. So again, primer, they can bind and they can result in the synthesis of 4 DNA fragments. And in the last slide, when 4 fragments, they are the target, in the third step, that will result in the production of 8 total fragments. तो अगर हम एक फ्रैगमेंट से स्टार्ट करें और आफ्टर 25 साइकिल हम ऑब्जर्व करें, तो हमारे पास जो डीएनए की यील्ड है वो अराउंड होती है 10⁻⁶ fold as compared to a first step. So huge quantity of DNA, it can be produced by using PCR. Now, in quantitative terms, we can say that if we start from a few fragments and up to 30 cycles, we can get huge amount of DNA fragments. So there is increase in exponential terms, that one segment it produce 2, 2 into 4 and 8 and so on. So PCR, it cannot only use to amplify the DNA fragment, rather it's also find its utility in gene cloning procedure. मिसाल के तौर पे अगर हम जो टारगेट डीएनए है जिसको हमने फोकस किया है कि हम इसको एम्पलीफाई करेंगे और इसको हम यूज़ करना चाहते हैं जीन क्लोनिंग पर्पस के लिए, तो हमारे पास ऐसी टेक्निक्स भी हैं कि जो प्राइमर हैं हम ऐसे डिज़ाइन कर सकते हैं जिसमें किसी ख़ास रिस्ट्रिक्शन एंजाइम की उसमें साइट इंट्रोड्यूस हम कर सकें। लाइक हम EcoR1 जो रिस्ट्रिक्शन एंजाइम है इसकी रिस्ट्रिक्शन साइट हम प्राइमर में इंट्रोड्यूस कर सकते हैं, और ड्यूरिंग द प्रोसेस ऑफ एम्पलीफिकेशन तो ये प्राइमर जो है इट बिकम्स द पार्ट ऑफ द एम्पलीफाइड डीएनए। अब इसमें चूंकि रिस्ट्रिक्शन साइट अवेलेबल है फॉर स्पेसिफिक रिस्ट्रिक्शन एंजाइम लाइक सपोज EcoR1, तो इसको कट करके सूटेबल क्लोनिंग वेक्टर में भी क्लोन किया जा सकता है। जैसा कि हम इसको एक्सप्लेन कर सकते हैं इस स्लाइड की मदद से कि हमारे पास एक टारगेट डीएनए है एंड दिस इज द रीजन दैट वी वांट टू एम्पलीफाई। आफ्टर डीनेचुरेशन स्टेप वी कैन यूज द प्राइमर दैट सपोज हैव साइट फॉर EcoR1 रिस्ट्रिक्शन एंजाइम। इससे क्या होगा कि डीएनए रेप्लिकेट करेगा एंड ड्यूरिंग द प्रोसेस ऑफ डीएनए रेप्लिकेशन दिस साइट target site it is incorporated into the amplified DNA fragment. No DNA fragment it is amplified with its respective restriction site goes for EcoR1 enzyme. With the help of this EcoR1 enzyme, now we can cut this restriction site and clone into a suitable cloning vector that is also fragmented with EcoR1. So in this way amplified fragments they can be used for cloning purpose. In addition to this application where we can use amplified DNA for cloning purpose, you are also well aware from our previous discussion that this technology it can also be used for the screening purposes, like the screening of genomic libraries or cDNA libraries. It can also be used for the diagnosis of different diseases and also in paleontology. So it also finds its utilization to know the past history where we can analyze the fossil record. So with reference to its application we will discuss at the end of this major topic.